certainly some things cannot be possible because the physics doesn't allow it, but in most cases when people say it's not possible, they have not tried hard enough to make it work.
Peter Hegemann and Georg Nagel have a lot in common. They're both German, and there's only 1 year's difference between them. They both have a passion for science, and particularly for biophysics, fields in which they've studied hard. Today, Peter Hegemann works in Berlin, where he's professor of biochemistry at the Humboldt University. Georg Nagel is at the University of Wurzburg, where he's professor of molecular plant physiology and biophysics. They may be 500 kilometers apart, but they've been linked together for nearly a decade by a protein much too small for the eye to see. After working for years with their respective teams, they joined forces in 2001 and showed that a rhodopsin protein that's present in green algae, an organism widely used by scientific researchers, made that organism sensitive to light. Georg Nagel then demonstrated the expression of rhodopsins in nerve cells of numerous species, including humans. Thanks to the two scientists, optogenetics was born.
This word just wants to say that with a gene you bring into, artificially, you bring a gene from one organism into your organism of choice, be it Drosophila or the little worm C. elegans or the mouse or so. So you bring in a certain gene and then you use light. And this is of course in the word opto. Yeah? You bring light and then you can change the behavior of the cell or the behavior of the organ, if, for example, the heart or so, which then will beat at a faster pace. Or you can also, of course, change then the behavior of your animal. And this was shown now in many different cases.
The public audience would like to hear that we can cure blindness or things like this. But first of all, optogenetic is an analytical tool or analytical technology.
This scientific discovery became a method designated Method of the Year 2010 by the journal Nature Methods. This technique has a wide range of applications and is used by numerous scientists around the world.
It's nice if you realize that you have done something which is not only important for a very, very limited number of people, or something which lasts for a longer time and many people can use it.
It was exciting because it was a new photoreceptor, and the sensory photoreceptors of this kind, rhodopsins, have never identified in a plant system. And so far it was first of all novel. It was not clear what would come out, and it took us, as you know, a little longer than we originally expected. So we thought we can identify it in 1 or 2 years, and it took us almost 15 years to find it. And this is how science is. You never know what will come, and I think this is also the attractiveness or the stimulating input you can give, or you can get from the basic science.
For basic research, it's a very, very good tool because it's a non-invasive tool. I mean, light is not invasive as long as it is not so strong that you get heated up by the light.
Using light, a natural and inexhaustible resource, for modifying cell behavior and perhaps for enhancing the body's capacity for autocure through internal stimulation. Thanks to the discovery of Peter Hegemann and Georg Nagel, many scientists today believe in this hypothesis, something that only few took seriously before. They showed that by keeping on looking, you end up finding what you were after, but that actually the most satisfying part is pushing back the horizons of knowledge, a virtuous circle, one that's difficult for the general public to appreciate, but which fascinates researchers.
You have to search if you want to find, so I have to prefer to search. And then I also, of course, like if I find something.
Inseparable. I mean, you cannot find without searching before. And if you do find something without having thoroughly searched, I think this is due to luck, and you may be spoiled afterwards. So I think it's both, and both can be motivating.
Maybe finding is more satisfying, but searching is maybe more interesting.
Find something is very nice, but I think research is very important.
I think it's a matter of character. If you will become a successful scientist, intelligent enough for most of the students, but only a few of them are ready to take risks and to work on totally novel issues.
After making this discovery together, Peter Hegemann and Georg Nagel now continue along their individual professional paths in fundamental research, each with their own teams of scientists, colleagues who come to share their experience, expertise, and so much more.
First of all, Peter is a good boss. I like him a lot, and I like to talk with him. And what you can get for him is that he likes to do cooperations with many different people. He's never, if you ask him, can I have support, and he will say, okay, I'm sending it to you.
Scientifically, he's very determined. He's very qualified. He knows what he has to do. He knows what he wants, and he's also very a long-term guy. He's not giving up.
Peter is also very open to constructive criticism or to comments himself. So it's not unidirectional that he would be telling other people this and that is the way you should do it. He's also very open to argument and to input from other people.
If you work with channel rhodopsin, everybody knows Georg Nagel. Because he basically started with his work in PNAS 2003. He started the whole field. So without his work, my work wouldn't be possible, and work of many scientists wouldn't be also possible at all.
I think his brain is always working. How we can do this, or can we do that, and maybe corporations. What can we do? What is the next step? And yeah, I think he's always thinking as a scientist, yes.
He has a lot of experience in this stuff, and we profit a lot of his experience. And this is very fascinating to see what an impact this work had that Prof. Georg Nagel did in the past, and yeah, to see how things changed. This is really a nice experience. Yeah.
While still keeping in touch, Peter Hegemann and Georg Nagel now have different objectives, one concentrating more on genetics and the other on optics and light. But for them, the two fields are clearly complementary.
Certainly the most important step was the identification of the photoreceptor and the demonstration how that works, and that this is a light-activated ion channel, and we have proved the concept together with Georg Nagel. And now we certainly work on modification, on changing the properties, on analyzing the details within the protein. This is based certainly on electrical measurements done in our lab, on time-resolved spectroscopy, and certainly on the structure that has to be provided by Osamu Noreki's lab in Japan. So we put all the information that comes from different labs and different experiments together to improve the system, to make better variants that we can then deliver to the neuroscientists that use it for neuronal network analysis.
It's very elegant to change the activity of a cell by light, but it should not be very intense light. And so one of the problems in the moment is that for the calming down of cells, for the inhibition of neurons, in the moment we only have the bacteriorhodopsin or the heterodopsin, which are light-activated ion pumps, and they need a lot of light. If we could generate a potassium channel or a chloride channel which responds to light, then we could do this with much lower light intensities. And this is something I would like to develop. There are some ideas, some initial experiments, and there will be a chance that we find a way also to inhibit cells by light, and we do not need anymore these very high light intensities which are needed in the moment.
The lives of Peter Hegemann and Georg Nagel are not limited to optogenetics, even if this discipline is now part of their shared DNA. At home, in their families, they continue to be keen scientists, but not only that.
No, that's not Peter. Peter is widely interested in so many things. Yeah, he likes to go sailing every year. We went to Croatia, sail there, and he goes mountaineering with his son, with students and other people. And he helps me in the garden. He has to help me in the garden. And I think he can also relax then when he works in the garden with me. And so he's not only a scientist, he's also a father and a husband.
I like sport. We do a lot of things together. Georg and I, we have a lot of things in common. We have the same generation, only 1 year apart. And so we really enjoy to do things together. And then we are different. So we are complementary in a way. Italian and German is the best match. Emotion with the rational. I mean, I'm curious about him, he's curious about me.
Peter Hegemann and Georg Nagel may have a lot in common as regards science and in their professional work, but the same is true in their private lives. Both have partners from the scientific world. Peter and Ute worked together for many years, and Anna Maria is a professor of molecular biology. Their children have taken different paths, sometimes more artistic or literary.
Although my parents both are like biology, physical, chemistry, whatever, I don't really of these, but they really support me, also, although I don't like the same stuff. I like everything that goes like in the creative kind of thing, music, dance, art, and I love languages, so I want to study. I would like to study French or Spanish or Portuguese, that kind of languages.
What I like on our father is that he never wants to influence us in what we would like to do or what we would like to study. I think he likes that I study science of biology,
it was
but
the last, sagt man, the last chance.
yes.
So
but
she is the youngest, and so it was the last chance to get somebody to
yes.
science.
Yes.
And for the two oldest child, it's the best. I think that's the latest is the one who is the science.